Hello everyone, welcome back to the series of solved previous year's ICAR PG examination, Plant Science, where we will discuss the questions on genetics and plant breeding. Today we will discuss the paper of 2020, so let's get started. Question number 1. What are the genotypes that will be included in the trial for the estimation of heterobeltiosis? Heterobeltiosis is the superiority of F1 over its better parent, so we require a F1 hybrid and the parental lines. This will be the correct answer. If we are using chicks, that time we will determine the commercial heterosis. Question number 2. Identify the correct statements about speed breeding. It is targeted to reduce the breeding cycle of a crop. It manipulates photoperiod and temperature to enhance crop maturity. It is done in open field conditions. No. It is done under controlled conditions or greenhouses. It was initially used to develop and grow the crops in space station. It increases the vegetative and reproductive phase of the plant. No, it actually reduces the generation time of the crop. So the correct statements are A, B and D only. Question number 3. An organism has haploid chromosome number 7. The probability that a sperm produced by it will contain all the 7 chromosomes whose centromeres were derived from the maternal homologue is, we know that n is equals to 7 and the probability can be out of two cases, either paternal or maternal. So the probability for getting only from maternal homologue is 1 by 2 raised to the power 7. That is option number 4, 1 by 2 to the power 7. Two genes are linked or not can be ascertained by, it can be tested by the chi-square test. F-test usually tells us about the equality of two population variances. T-test is a measure of a statistical difference. Z-test is used to determine whether the population means are different when the variance is known and the population is large. Question number 5. When two self-incompatible but cross-compatible parental lines are interplanted, the seeds harvested from both the self-incompatible lines will be, we know there are two self-incompatible lines, parent 1 and parent 2, but they are cross-compatible with each other. So when they are crossed, we have the hybrids developed. The correct answer will be option 3, the hybrid seeds will be developed. Which of the following stages of cell division is primarily arrested by treating with colchicine used in polyploidization of plants? So the stage arrested is the metaphase. The colchicine interferes with the formation of spindle fibers, thus retarding the division. Question number 7. The tag for breeder seed is issued by, it is issued by the concerned breeder of the variety. Question number 8. What is the probability when both parents with blood type A and normal vision produce their first son who has blood type O and is colorblind? So we know if the son is getting blood type O, the parents might be heterozygous, which tells us that the probability of getting IO is 1 by 4. If the son is colorblind, the mother may be the carrier and the father is normal. So which implies that there is 50% chances of getting colorblind. The total probability comes out to be 1 by 2 into 1 by 4, which is 1 by 8. So the correct answer is option 3, 1 by 8. Question number 9. A cultivar developed through population improvement approach can be maintained indefinitely with proper identity through, so they can be maintained by random mating in isolation. Which of the following is incorrect statement about hybrids? They can be produced only in cross-pollinated crop species. This is the wrong statement because they can also be produced in self-pollinated crops by the use of heterosis and apomixis. Question number 11. Match list 1 with list 2. Mitochondria we know that they help in oxidation of carbohydrates, lipids and proteins. Then we have endoplasmic reticulum. It provides the passage for mRNA from nucleus to cytoplasm. Lysosomes, they help in digestion of intracellular substances and foreign materials. Golgi complex, it is used in packaging of proteins, lipids and phospholipid for transport to other cells. So the correct answer would be A2, B4, C3 and D1, that is option number 2. Question number 12. Match the genotypes with the population. Inbred lines, we know they are homozygous as well as homogeneous. The three-way cross hybrid, individually they are heterozygous. Also the population wise also they are heterogeneous. Single cross hybrid, they are heterozygous and a population is homogeneous. Multi-lines, they are homozygous individually whereas the population is heterogeneous. So the correct answer would be A2, B3, C4 and D1, that is option number 4. Question number 13. The genome of triticale is designated as, so from here we can see Triticum turgidum has gametes AB and cecale serial has R, which develops ABR, and applying the colchicine we have the chromosome AA, BB and RR. So the correct answer is option 4. Identify the correct order of activities followed by testing and release of crop varieties. First one, we know the stational trials are conducted. Then the multi-location testing is done, followed by the varietal identification. Then the release and notification occurs. After that, seed multiplication and distribution is done. So the correct order is B, A, C, D, E. Question number 15. Stalk 6 is a haploid inducer line of. The correct answer is option 2, maize. Haploid inducers, they are able to induce embryos of paternal or maternal origin. Starting from interior to exterior, arrange the following in correct order. So from the interior, we have first nucleus. Then we have endoplasmic reticulum. After that we have plasma membrane, followed by the periplasmic space. Lastly we have the cell wall. So the correct option will be BC, BEC and AD. Which one of the following is not an introduced variety of wheat crop? So it is laticale. Question number 18. Match list 1 with list 2. Double cross hybrid, we know that they are crosses between F1s of A and B and C and D. F1 hybrid is a single cross hybrid. Average heterosis is known as mid-parent heterosis. Economic heterosis is also called standard heterosis. So the correct answer would be option number 2, A3, B4, C2 and D1. Given below are two statements. Hybrids are heterozygous and homogeneous. Pure lines are heterozygous and heterogeneous. Choose the correct answer from the options given below. So we know that the hybrids, they are heterozygous and the population is homogeneous. It's correct. Whereas the pure lines, they are homozygous as well as homogeneous. So the statement 2 is incorrect. That is option number 3. Statement 1 is correct, whereas the statement 2 is incorrect. Question number 20. Which of the statements below do not stand true about Convention on Biological Diversity? CBD deals with all biological resources. One of the objectives of CBD is the conservation and sustainable use of the component of biodiversity. It affirms the sovereign rights of the nation over its genetic resources. It is an instrument for harmonizing phytosanitary measures for international trade. CBD provides a model for securing protection for plant breeding rights and plant varieties. Out of these, only the A, B, C statements are correct and the wrong statements are D and E. So the correct answer will be option number 4, D and E only. Which of the following plant organelles is considered as semi-autonomous? So out of these, the correct answer is option number 2, mitochondria, because they have their own genetic material and can synthesize proteins. Also, chloroplast is also known as the semi-autonomous organelle. Question number 22. Given below are two statements. Statement 1. When one dominant allele of one gene and a recessive allele of another gene are present on one chromosome and their opposite alleles are present on the homologous chromosome, the arrangement is called cis-heterozygote. So this statement is depicted like this. These genes are translocated, so this term or this arrangement is called trans-heterozygote. Whereas the statement 2 says, when one dominant allele of two genes, they are present on one chromosome and the corresponding recessive alleles are present on the other homologous chromosome, the arrangement is called cis-heterozygote. So this is known as cis-heterozygote, whereas this arrangement is called trans-heterozygote. This says that the statement 1 is incorrect, whereas the statement 2 is correct. Question number 23. How many linkage groups are present in Zea mays? So 2n is equal to 20, which says that the number of autosomes in the haploid cells will be 10. This implies the linkage groups, they are 10 in maize. Question number 24. Given below are two statements. Kosambi's mapping function considers the absence of interference. Haldane's mapping function considers the presence of interference. Both the statements are incorrect because Kosambi's mapping function, it assumes certain degree of interference, and Haldane's mapping function, it assumes there is no interference between crossovers in the meiosis. Question number 25. A crop variety enjoys legal protection in India provided it should be registered with the Plant Protection Varieties and the Farmers' Rights Authority. Question number 26. In a cross, what is the probability that any particular gamete from the female parent will be capital A small b, capital C small d and capital E? So if female parent is considered, we always take the parent 1 and we can follow the branching technique or the direct cross method. From this we have 8 different gametes and the probability of getting capital A small b, capital C small d and capital E is 1 by 8. So the correct answer would be option number 4, 1 by 8. Question number 27. Arrange these conventions and treaties in ascending order of year they entered into force. WTO-TRIPS, it came in 1990. CBD came in 1992. Geographical Indication or the GI Act, it came in 1999. Then we have the Nagoya Protocol, it came in 2010, and the International Treaty on Plant Genetic Resources for Food and Agriculture, it came in 2001. So the correct order would be A, B, D and EC, that is WTO TRIPS, CBD, GI Act, International Treaty on Plant Genetic Resources of Food and Agriculture, and then the Nagoya Protocol on Access and Benefit Sharing. Question number 28. In Hershey's and Chase experiment, phosphorus and sulfur were used because out of these statements, only C and D are correct because phosphorus is present in DNA and sulfur is present only in protein. Selective labeling of DNA and protein can be done as phosphorus 32 and sulfur 35. Other statements, they are wrong because they are absent in RNA. Then they are present in DNA and protein. This is wrong. They both are available in gaseous form. This is also wrong. Only the correct statement is C and D. Question number 29. The first product to be tagged with geographical indication in India is, it is known as the Darjeeling tea of West Bengal. Question number 30. Given below are two statements. Cleistogamy and chasmogamy favor self-fertilization. Male sterility and self-incompatibility do not favor cross-fertilization. The statement 1 is correct and the statement 2 is incorrect. Question number 31. To estimate the genetic diversity of germplasm collection, a technique involving multivariate analysis based on ordination giving graphical representation is called. It is known as principal component analysis. In a sexually reproducing organism, the chromosome number in the progeny does not double even after fertilization. The reason is that the male and female gametes contain half set of somatic chromosomes. That is why the chromosome number does not double after fertilization. Question number 33. Cell theory is put forward by, it was given by Scleden and Swan in the year 1839. The allelic combination of the variety 1 is this and has the phenotype of 6 yield units. If the allelic combination of variety 2 is this and has 4 yield units, what would be the yield unit and heterosis unit of the F1 if there is complete dominance? For F1 we have capital A small a, capital B small b, capital C, capital D, capital E, small e. This is the F1. For variety A we have 6 yield units. For variety B we have 4 yield units. If F1 units they are calculated, there are 5 dominant genes, which means there are 10 units of yield. The yield units will be 10 and the heterosis, that is F1 minus the better parent, 10 minus 6 is equal to 4. That is why yield unit comes out to be 10 and the heterosis is 4. The correct answer is option 3. Question number 35. Given below are two statements. Higher the degree of cross-pollination in crops, more will be the isolation distance requirement. Self-pollinated crops need more isolation distance like cross-pollinated crops. The statement 1 is correct and the statement 2 is incorrect because self-pollinated crops do not need more isolation distance. Question number 36. The gene capital A, capital B and capital D, they are present in one chromosome in coupling phase of linkage. One of the double crossover gamete has capital A, small b and capital D gene combination. The gene which was present in the middle of the sequence is, we can see in coupling phase, if we do the double crossover, we have capital A, small b, capital D and small a, capital B, small d. These are the recombinants. And in coupling phase, we have the parent, capital A, capital B and capital D and small a, small b and small d. For double crossover, we are given capital A, small b and capital D. Here we can see the gene B is changing and the capital A and capital D, they are same. So that is why the change present, it is seen in B. So therefore, the middle of the sequence will have B gene in them. Question number 37. As per Seymour Benzer, a unit of mutation muton and a unit of recombination recon respectively represent, so out of these statements they represent only single nucleotide is muton and adjacent pair of nucleotide they are recombinant units, that is recon. Here we can see cis-tron is the gene unit, recon adjacent pair of nucleotides and muton a single nucleotide. Therefore only the correct answer is B. Given below are two statements. Inbreeding depression is the loss of vigor and fertility due to homozygosity of deleterious recessive alleles. Inbreeding depression is the loss of vigor and fertility due to heterozygosity of recessive alleles. So the statement 1 is correct and statement 2 is incorrect because inbreeding depression is only due to the homozygosity. Which one of the following is not a nonsense codon? So AUG, which is also known as start codon and codes for methionine, is not a nonsense codon. Question number 40. The yield of inbred A is 40 quintals per hectare. Inbred B is 20 quintals per hectare. Czech hybrid yield is 45 quintals per hectare and the yield of hybrid is 50 quintals per hectare. The standard heterosis will be, so the standard heterosis is determined by the formula F1 minus the Czech upon Czech variety whole multiplied by 100. So by putting the values in the formula, we get 11.1%. The correct answer is option 2, 11.1%. Question number 41. Given below are two statements. Cis-trans test can assign mutant alleles to the same or different genes but cannot estimate the map distance. Recombination frequency can be used to estimate map distance between the mutant loci. So both the statements are correct. At the minimum, how many years of multi-location testing is required for a conventionally bred genotype to be released as a new variety? It should be up to 3 years. Question number 43. In a crop, genes capital C and capital P, they are required for the development of color in the flowers. The white flowers are produced in the absence of either or both the genes. What will be the percentage of colored flowers in the offspring of this cross? So of this cross, if we develop the gametes, there will be capital C capital P, small c capital B, small c capital P and small c small p. If selfing is done, we get 50% of the genotypes which have capital C and capital T present in them. If they are not present, then the white flowers will be produced. So the correct answer is 50%. Question number 44. Match the lists of theories with the scientists who proposed them. The cell theory was proposed by Schleiden, Swan and Vercoe. Chromosomal theory of inheritance was given by Sutton and Boveri. Divisibility of gene was given by Clarence P. Oliver. One gene, one polypeptide theory was given by Vernon Ingram. So the correct answer would be A2, B4, C1 and D3. That is option number 2. Question number 45. The possible number of triplet codons from 6 different nucleotides in the DNA molecule is n is equal to 3 and we have given 6 different nucleotides. So it will be 6 raised to the power 3. That is option number 1. So that was it. Thank you.